All right, now we're going to go in big picture and look at what happens in meiosis. So first of all, remember that when we're talking about meiosis, we're talking about starting with a diploid germline cell. Okay, so we've got a 2N cell. During interphase, the DNA is going to replicate. Okay, that's going to happen during S phase. And after S phase, we are going to have chromosomes that are pairs of sister chromatids. So remember what I told you last lecture, that we count chromosomes by counting centromeres. So prior to DNA replication, we had 2 centromeres, so 2 chromosomes. After DNA replication, the sister chromatids are held together at a common centromere. So we still have 2 chromosomes. So our cell is diploid, but each chromosome is a pair of sister chromatids. Now, at the end of G2, this diploid germline cell is going to enter meiosis. Now, meiosis is broken into 2 big phases. There are 2 separate rounds of cell division in meiosis. Big picture, the goal of meiosis 1 is to separate the homologous pairs of chromosomes. So after meiosis 1, we have 2 haploid cells. But notice each haploid cell is still a pair of sister chromatids, or each chromosome is still a pair of sister chromatids. So up here at our original parent cell, it was diploid. We had 2 centromeres, 2 chromosomes. After meiosis 1, we only have 1 chromosome per cell, 1 centromere, 1 chromosome, but each chromosome is a pair of sister chromatids. Well, the goal of meiosis 2 is to separate the chromosomes so that at the end of meiosis 2, we have 4 haploid cells. Okay? Each cell only has 1 chromosome. Right? Now, there's a lot of other stuff that happens, especially in meiosis 1, but that's the big picture overview, all right? So we start with a diploid cell at the end of meiosis 1. We have 2 haploid cells, and at the end of meiosis 2, we have 4 haploid cells. So meiosis is also known as reduction, division, and what's being reduced? The number of chromosomes. Okay, so going from a diploid cell to haploid cells. Now, in meiosis 1 is subdivided, but notice it's the same order of subdivisions as it was for mitosis. So prophase 1, metaphase 1, anaphase 1, telophase 1. So in subsequent videos, we're going to talk about the specifics of what's happening in these phases, but this figure just gives you the big picture of what's going on in each phase. Okay, some really interesting stuff happens in prophase 1, and then some really important stuff happens in metaphase 2 that sets up what happens in meiosis 2. That's really important for genetic variability in populations that reproduce sexually. So let me go back here. So remember, the goal of meiosis 1 was to create 2 haploid cells, but each chromosome is still a pair of sister chromatids. Well, in meiosis 2, the goal is to separate the sister chromatids. Meiosis 2 is essentially mitosis. Okay? And what I mean by that is the same series of events that happens from prophase 2 all the way to the end of cytokinesis is the same series of events that happens from prophase to cytokinesis in mitosis. Okay? Now, don't get confused and think that the DNA is replicated again between meiosis 1 and meiosis 2. It's not. There's only one round of DNA replication in meiosis, and that happens in S phase prior to meiosis 1. All I mean by saying it's essentially mitosis is that the chromosomes are going to line up on the metaphase plate, and then the chromatids are going to be separated. Okay, so those series of events are essentially the same. And again, we'll talk about the details of these processes